Good. Then let's start. Good morning, everybody. It's a pleasure to be here for me. My name is Carsten Ma. I'm a research group leader and deputy director of the Institute of Computational Biology at the Helmholtz Center in Munich. And it's a great pleasure for me to be the host today for Maria Colomé-Tatché giving a talk in our SCOG single-cell omics Germany network event series, a really great series with great speakers. And today it's Maria on single-cell computational epigenomics. Let me briefly outline Maria's career. Maria studied physics in Barcelona, in Spain, where she also grew up, I guess. She moved to Paris for a while, then also came to Germany for a short trip, and then went to Groningen to start her own little lab there. And then, scientifically, she also moved from physics to computational biology. And I took the time to dig a bit into your publications, Maria, and I'm impressed by the fact that you managed to publish in physical review letters, right? That's basically the best you can do as a physicist. I didn't get there, but you did, obviously, with the work on parametric excitation of a 1D gas in integrable and non-integrable gases. Remember that?
That was my past life.
10 years back. So Maria, in between, switched a little bit topics. So she worked on plant epigenomics in particular. And now since she joined the Helmholtz Center, that was 4 or 5 years ago, Maria?
4.5, more or less.
She's now looking a lot into the analysis of epigenetics, in particular now, since we have the data in single cells. And I guess that's also what Maria is going to cover. Maria is a super great colleague at the Helmholtz Center. We enjoyed interesting projects and retreats, and I'm very much looking forward to that talk, Maria.
Thank you very much, Carsten, for a very, very nice introduction. Wait, I'm trying to share my screen. Do you want to continue? Continue. There we go.
While you do that, let me tell you that, as usual, if you want to ask a question, please, I mean, we have a 15, so this will be a 30 minutes talk, and at the end we have a 15 minutes question round, so please take notes about the questions you want to ask. Put a cue or a question into the chat so I can call you at the end, or raise your hand in this reactions thing at the bottom right.
Good. Thank you so much, Carsten. So now I hope everybody can see my slides. I hope this works well. My computer, my usual computer decided to restart this morning and saw a lot of updates. And at 9, it told me it would take 45 minutes. So I have it here next to me and it's still updating. So I'm using an old computer, which I hope, let's say thanks Dropbox for keeping all my files, and I hope it all works fine. I though cannot put a laser pointer because it's an old computer, but I hope you can see my mouse.
We see your mouse, yeah.
Okay, so thank you very much for inviting me to give a talk today and to present my work to the SCOG community. I still remember when I joined ICB. Indeed, it was when Fabian was planning to create this SCOG network that was actually initially submitted as something else, and then we got this very nice idea. So it's so nice that, especially now with the pandemics, this can help us so much stay connected. Okay, so I will talk to you today about the analysis of single-cell epigenomic data, mainly in general, when, let's say, when I have collaborators and they tell me we have single-cell data. This always means single-cell transcriptomics data. But we actually do not realize is that, or yeah, we start to realize now is that with the single-cell transcriptome, we miss all this regulatory information that is sitting between the expression of the genes and the genome itself, which is the epigenome, which is what we are interested in my group. So let's say with the single-cell RNA-seq alone, we miss a big part of this regulatory component of the single cell. And it's also obviously a gene-based approach because we can only measure the genome at the position where there are genes which are being expressed. So if we instead measure the epigenome, this is a whole genome approach where we get information both at the genes, but also at all the non-genic regions of the region of the genome, and therefore also the transregulatory and cisregulatory information. So nowadays, as you very well know, there are many different single cell omics layers that we can measure for the same single cell or for different populations of single cell. We have got, of course, the genome with the single-cell DNA measurements. We have, of course, the transcriptome with the single-cell RNA-seq, and there are arguably two different layers of epigenomic information, which are the ones which are the most measured at the moment, which is the single-cell DNA methylation with the use of single-cell bisulfite sequencing that has sometimes other names, but it's always a similar principle, or the chromatin architecture, so whether the chromatin is closed or open, mainly with the use of the single-cell ATAC-seq nowadays. So especially with single-cell ATAC-seq, the amount of data which is produced is really exploding, because now we have Kinnex, for example, that has released a kit that allows us to do that very easily. So we argued in the group, if we are interested in studying how these layers of regulation go from the genome to producing different transcriptomes, what we need to do, it is to generate a good platform for the study of single-cell epigenomic data. So what we did is that we generated a platform that we called EpiScanpy for epigenomic single-cell analysis in Python. I'm sure you're nearly all familiar with SCAMPy, which is the analysis platform for single-cell transcriptomics that was developed in Fabian's group. So we obviously built on top of that pipeline, but generating it for the analysis of both single-cell DNA methylation, single-cell ATAC-seq, the integration with the single-cell RNA-seq via the SCAMPy branch. And of course, we already built it in a very modular way that will allow us then for a quick multi-omic data analysis once we have the different omics layers for the same cells. So I will just go quickly over how this pipeline works. The first step is the feature selection. So why do we need to select features? When you do the single-cell transcriptomics, you don't even think about the features, isn't it? You just have your genes, have the expression of the genes. So the genes are your features, but you never really thought about it. But when you do single-cell epigenomics, you have information everywhere in the genome, and you cannot really summarize it at the base pair level, because that would be way too huge and we would not be able to analyze this data. So there are different approaches now, and in the literature, people have taken different ones. We decided in EpiScanpy to be as general as possible. So we can either take the whole genome, and here you just make windows, equally sized windows along the genome, you consider everything. Or you can say, no, I have really like a genome annotation that I'm interested in. I want to know the openness or the DNA methylation at the promoters, for example, or at the gene bodies, or I want regulatory information. So I want openness or DNA methylation at transcription factor binding sites instead. Or of course, if you have open chromatin, you can say, no, I have my peaks and I want to know what's the openness of these peaks. So then what we do, of course, is that we take whatever genomic annotation or genomic features that the user is interested in, and then we can build for every one of these features a summary of how open that feature is or how methylated that feature is, depending on we are working with the single-cell DNA methylation or single-cell ATAC-seq. And then we can construct, let's say, as many count matrices as feature spaces we are interested in that will contain the cells and the features with their quantification. Then, of course, we do some pre-processing, as always, some QC of the data, and then we use this nice, clean data set with the cells and the features and their openness or DNA methylation here with these colors to measure how similar, to kind of define a distance between the cells based on their similarity, based on their epigenome. And then we have what we call a neighborhood graph that then we can use for many, many downstream applications, for example, for visualization with the UMAP. We can, of course, also do clustering. We use Louvain clustering in our case. Once we have these clusters, we can ask what is differentially methylated or differentially open between the clusters. We get to a certain number of markers that then we can use to go back and say, okay, then this cluster is most likely whatever cell type. There are, of course, situations where we don't want clusters, but we want a continuous representation of the data. So we can also do pseudotemporal ordering. And we can also do data integration if you have data sets that have a batch between them. So I will give you two short examples on how we can apply these techniques. So one of them is on single-cell DNA methylation, and the other one is on single-cell ATAC-seq. So for single-cell DNA methylation, this is all publicly available data. This is a paper from the Joe Eckert group that has more than 3,000 single nuclear methylation sequencing cells. It has a very, very, very low coverage, so less than 5% of the cytosines have information. But so what we can do there is that we can get the methylation status Did I say it was brain? I didn't say it was brain. It is brain, as you can see here in the title of the paper. Yeah, so sorry. Then what we do is that we get the methylation status of every one of the individual cytosines. We build count matrices, and we can build them, as I mentioned, for different features. So we have a different set of count matrices. And for example, here on this example, I'm showing to you if we have used the enhancer count matrix, which means that we have quantified DNA methylation, not everywhere in the genome, but only at the positions of the enhancers, okay? So our count matrix is cells versus enhancers, and we can then use this count matrix, for example, for this low dimensional data representation and for doing clustering. And as I mentioned, we can use different feature spaces. So enhancers, promoters, with windows and gene bodies here, for example. Here we took CG methylation. So something that one can do is that one can start then asking questions, how different the cells get grouped if I use the enhancers versus the promoters, for example. And you can see this in this plot here. You can see that in general, a cluster in the promoters is also a cluster on the enhancers. So there is not too much differences, whether you look at the enhancers or the promoters, but there are some cases where indeed we can see some cluster, like for example, here in the promoter cluster 12, that gets nicely split into two separate clusters in the enhancers, and these are the ones here on the UMAP. This is not our data, so we didn't dig any deeper, but this is something that we find really, really interesting. If you have your own data set and you're interested on, what is really the determinant of cell identity. And then, yeah, sorry, we also realized that in the case of DNA methylation, the enhancer feature space actually gave us much tighter clusters, so it seemed to be somehow better capturing the cell identity. Then until here, these are groups of cells with colors, so we don't know what they are. So what we obviously want to do is that we want to do cell type identification. We want to ask, okay, what are these cell types? And to do that, what we do is that we want to identify markers by ranking the differentially methylated features between the clusters. So we ask, if I compare these two clusters, which ones were the features that were the most highly differentially methylated between the two? And we can do that using any of the feature spaces that I told you that we can build. But of course, if we use the promoter feature space, is the one which is then more kind of straightforward, because you get names of genes which promoter was very differentially methylated. So you can kind of link it easier to the gene names. So here what I'm showing to you is the same UMAP as before, but with very low resolution, where we said, no, we just don't want so many clusters in our data, just split it into two. And then we asked which ones are the top most differentially methylated promoters between the two. So you can see this is the orange cluster and the blue cluster. And as Carsten said, I'm not a biologist, but we have a lot of people here on campus which are very good on brain biology. So we show them this list and they told us, oh, look, there is NeuROT2 here on the list. And NeuROT2, indeed, if you plot it on top of the UMAP, you see that it is high methylated and low methylated. You can also plot it as a violin plot. And since NeuROT2 is known to mark inhibitory neurons, then we could come to the conclusion, okay, then one of the two clusters is the inhibitory neurons and the other one is the excitatory neurons. And we did it for this very low resolution situation where, you only have the two clusters to kind of start delineating what big cell types, big kind of most important cell types do I have. But then of course, you can also go into any one of these separate clusters and do the same procedure. So then for every one of the clusters, you get a list of genes. From the list of genes, you can talk to a biologist that will tell you, no, this is then most likely this cell type or that cell type. And then we can go to this cell type annotation. Then, as I mentioned, we can do the exact same thing with single cell ataxic data. This is again publicly available data from the Greenleaf lab, where they have profiled PBMCs along differentiation. So this is 10x data, and we also have a very sparse coverage. So we have on average only 13,000 reads that fall into features per cell. So it's pretty sparse, but again, like with DNA methylation, we can construct the peak accessibility count matrix. We can also construct different count matrices, but here we only went with the peaks. We can use this for the low dimensional data representation and the clustering. Here we have quite a few cell types that we can represent with these clusters. But obviously, oh yeah, sorry, we can do the same trick as before. We can here ask which ones, sorry, what you see here is 0 very close peak, 1 very open peak. And for this main split on cell types, we have 2 markers that we've shown here that are very open for one of the cell types and not the other. So, I mean, obviously, when we do cell type identification, we don't do it with 2 markers, we do it with a lot, but you can use this kind of representation and visualization to then get, the peak ID. On EpiScanpy, you can then use a function which is called find gene that tells you which one is the gene, which is the closest to this peak, which is so differentially open. And then you can go from these gene names to the cell types. But yeah, sorry, what I was saying, as you know, blood is a continuously differentiating tissue. So more than representing it in separate clusters, it's more interesting to represent it as a continuous differentiation. And for that, we can do diffusion pseudotime without RNA-seq data, only using the ATAC-seq data. And you can see diffusion time 0 all the way to 1. And we can annotate it, and we see that we have the progenitors here. And then we have the T branch, B, basophil, and myeloid. So we can see the different cell types differentiating. We have, of course, benchmarked EpiScanpy to other tools. This is a very busy slide, but you don't really care about all the details. The top method here listed is EpiScanpy. We have tested it on 4 different data sets on its ability to really discriminate cell types. So we use the adjusted run index to quantify the goodness of the clustering. And what you can see is that EpiScanpy comes up top in all the data sets except of one. Well, it happens, you're not always the best, but we are happy that we perform always consistently on top, let's say, of the majority of the methods. And what we see that we are very also happy about is that, of course, we are Python-based, we're very fast. So, for example, on this data set here, we have more than 80,000 cells, but we still can calculate a result in 18 minutes, which is really good if you want to explore the data. If you want to change something here, something there, and see how does it look again. You don't have to wait 3 days every time. So we also think this is very nice. Okay, so until here, I've told you about EpiScanpy for the analysis of single-cell DNA methylation, single-cell ATAC-seq, can do all the usual downstream that you may want to do, given any input that you're interested in. We scale very nicely to large data sets, and we perform well to other methods. And this paper has just been accepted in Nature Communications. So yeah. Okay, so until here, just one data set. How do I do to analyze one data set? But the thing is that especially now with the advent of the 10x technologies, we are producing more and more and more data sets. And very often it happens that you have an interest on a given tissue or organ, or you are producing your own data. And then you realize that there is another lab that has also produced this data. And you wonder, why not use, if I have 100,000 cells and they have 100,000 cells, why not have 200,000 cells instead? So how do we do to integrate and remove all the batches that come with data integration with single-cell ATAC-seq? So the thing is that currently there is no method for the integration of single-cell ATAC-seq data sets, but there are arguably a lot of methods for the integration of single-cell RNA-seq data sets. So what we thought, together with Fabian's group, we thought, let us evaluate how well the single-cell RNA-seq data integration methods perform on single-cell ATAC-seq data instead. So we didn't want to make it easy, so we didn't want just 2 small data sets to integrate. So we chose 3 different data sets from 3 different kind of technology platforms. The 3 of them, of course, need to be the same cell type. So the 3 of them are for brain. The first data set, it's a combinatorial indexing data set from Kuzanovic et al., where they have 18,000 cells. The second data set is from mouse cortex from 10x. And the last data set is around 80,000 cells from, again, the Joe Ecker and the Bing Ren lab. So 3 data sets for brain. Now, what do we have? We have 3 matrices, isn't it? We have 3 count matrices with the cells and whatever feature I'm interested in the genome. And in order to integrate them, the first step in general is that you just concatenate the matrices. So if I had, 5,000 cells, 5,000 cells, and 80,000 cells, I just concatenate the matrices and now have a matrix that has 90,000 cells. But this is actually straightforward, obviously, in the single-cell RNA-seq, because on my columns, I have gene expression. And the genes are the same for all the data sets. But when I do ATAC-seq data, well, I don't really have the same open chromatin. So how do I actually make sure that when I concatenate the matrices, the columns have the same information? So this was a little bit of a point that we discussed a lot. The first approach that we took was to take windows, because of course, if you have the same genome and you split it in windows, it's the same windows in all of them. So the red, the green, and the blue data set, we have windows, it's not going to be a problem. But then this came a lot with the discussion with the people that come from the RNA-seq field. And they were like, well, come on, why don't you instead use what people have been calling a gene activity, which is you take the openness at the promoter and the gene body. And this gives you an indication of how active the gene is. And this seemed to be a good idea because since we are using single-cell RNA-seq integration methods, the best is if we render our single-cell ATAC-seq data to a format that looks the most similar to the single-cell RNA-seq. So we also did that. We defined this gene activity, which is again easy because the genes and the promoters are the same position for all the cells. So we again can very quickly build that. But then we come more from the epigenomic fields and we were like, come on, well, the information is in the peaks, isn't it? So wouldn't it be nice to use peaks instead? But this is a tricky one because the peaks are definitely most likely not be the same ones in every one of the data sets that you're going to use because you call peaks in every data set separately. So the peaks that are, let's say, are kind of the same one are still not in the exact same positions, and there are also some peaks which are going to be unique to the data set. But what we did is that we kind of took the union of the peaks and we went with the union of the peaks for integration. So now we have three matrices that we can indeed concatenate because they have the same features, and we just ask, how does the data look like on these concatenated matrices? So here you can see the green data set, the orange data set, and the blue data set, which are the three ones here. And you can see that they are separate on the UMAP. So you can see the batches by eye. This is on the top for the Windows feature space, for the peak feature space, and for the gene activity feature space. And what you can also clearly see is that the gene activity feature space is not what you should probably be using, at least for embedding in the single cell ATAC-seq field, because you really lose a lot of the information which is present in the data. So, okay, let's integrate these data sets using different batch removal techniques. This is a super long list. I don't expect you to look at all the names there. These are different methods that we chose together with Fabian's group for the benchmarking. These are single cell RNA-seq integration methods. And we, of course, need to quantify how good are they at integration. So we came up with different measures, both for batch correction and for biological conservation. Why do we need both measurements, both kind of metrics? So imagine you have two batches, the blue batch and the yellow batch, and you want to integrate them. You could have a method A, that if you look at the blue and the yellow mixture is excellent. It has really done a very, very beautiful integration of your two data sets. But then if you look at the cell types, and the cell types here are the circles, the triangles, and the stars, it completely went wrong. It did a great job at batch correction, but a terrible job at conserving the biological information present in the data. So the cell types are not separate. What we want is a method like the method B that both can mix the colors, so it has a good batch correction, but it can also preserve the biological information present in the data, so it can keep the cell types separate. So this is what we did. Super busy, don't even look at it. But this, what we did, we have an overall score, which ranks to you the best to the worst method. And this overall score is a combination of this batch correction score and this biological conservation score. And these are just the individual metrics that we measured. Interesting thing, this one here says unintegrated. So this is just concatenating the three count matrices. And you see that there are quite a lot of methods that, well, you think I may be better using the unintegrated one instead of applying this integration method. But there are some methods that perform better. So let's remove the middle ones that we kind of don't care. This would be an example of a good integration. Harmony has a good compromise between a good batch correction, but also a very good biological conservation. So you can see the three batches are well mixed, and the cell types are also nicely differentiated among them. It's easy to show you examples of things that didn't work out. For example, saucy here, didn't work out very well. And the interesting thing is that here I show you the feature space, so Windows peaks, and then genes. So it looks like if you go with the gene feature space, you're definitely not doing so well. You find a result which is somehow worse than really using just the unintegrated ATAC-seq result. So use gene activity for other things like identifying cell types and so on, but definitely not for an embedding and not for integration of data. So, okay, the summary of this part, we know that integration of large ATAC-seq datasets is actually a challenging task. We have used different single-cell RNA-seq data sets for that. Sorry, not data sets, single-cell RNA-seq methods for the integration of single-cell ATAC-seq data. We see that definitely pigs and windows are much better at integration than the gene feature space. And there is, of course, a very large trade-off between batch correction and loss of biological information, because I didn't show to you, but there are some methods that were very good. Come on, at the, you see, were very good at the batch correction, but then were pretty bad at the biological conservation. So it kind of made a blob with all the cells. They were very nicely integrated, but they didn't preserve the different cell types. And as I mentioned, this has been a very nice collaboration with the Fabian Theis group. It has been led by Malta and Maren, the first two authors. They were interested on benchmarking the integration of single-cell RNA-seq, where they really used a lot of different data sets, and we contributed with the integration of the single-cell ATAC-seq. So now I will tell you just a tiny little bit of a last project that we started very recently in the lab, but I am kind of very excited about, which is calling copy number variations from single-cell data. So, okay, you have a bunch of cells and you do single-cell measurements. And of course, what we know is that when you map the reads on the genome, so here what you see, every dot is kind of a summary of how many reads are mapping on that position in the genome, sometimes we see these jumps on the data. And this is if we have some copy numbers present in the data. So let's say I have an arm of a chromosome that is trisomic instead of being disomic. Of course, since I am sequencing the DNA in that position of that specific single cell, what I see is that when I map the number of reads on that position in the genome, I see much more reads mapping there, because I basically have 1.5 more substrate to sequence. It kind of makes sense. So some years ago, before I go into that, it's very nice that we can do that from the single cell sequencing data, because until now, let's say if we wanted to study single cell, the karyotype of single cells, we either needed these very classical measurements of karyotyping, which arguably gave these beautiful pictures of these karyotypes of the cells. I think this is HeLa. But they are either limited to dividing cells to obtain these beautiful images, or they can only profile a small number of chromosomes, because they use fish probes. So then you can only put as many fluorescence probes as you can. If you don't care about the single cells, you only care about the bulk, then there are a lot of methods indeed that can be used, ACGH, exome sequencing, whole genome sequencing, etc. But this just gives you an idea of the population, not how much heterogeneity I have. So it's really cool that with the single cell sequencing now we can also access this extra layer of information about the cells. So some years ago, this was back in 2016, in the group we developed a method that we called AneuFinder for calling copy number variations from single cell whole genome sequencing data. Okay, so we don't care about the transcriptome or the epigenome, we really measure the genome of the individual cells. So this is just like the standard pipeline. You have your cells of interest, you do a single cell genome sequencing, and then you map the reads to the genome. Here you see a few chromosomes, and what you can see is that there are some chromosomes that suddenly have much less reads. So arguably there has been a loss of a chromosome, and some that have more reads, so probably a gain of a copy of a chromosome. What we did back then with Aaron was my PhD student in the Netherlands, we bin the genome and we counted how many reads are mapping in the genome. And then if you plot that as a histogram, what you can see is that you have like different distributions there that correspond, for example, to the regions which are disomic, the regions that have had a loss, so that have half a number of reads, and the regions that have had a gain, so have like 1.5 more reads. So we can then fit distributions, a mixture of distributions to this data. And back then we used the hidden Markov model to model this process that tells us how likely it is that, let's say, if I am in a monosomic bin in the genome, the next bin will also be monosomic, or that if I'm monosomic, the next bin will be disomic, or if I'm disomic, the next bin will be trisomic. So this is what the hidden Markov model does. And we use these negative binomial fits to model these emission distributions to generate these numbers here. We used the Baum-Welch algorithm, and then we fit that and we found which one is the most likely hidden state for the whatever cell that we are considering. This was single-cell DNA-seq. This was 2016, but since then we have so many more things that we can measure at the single-cell level. And single-cell DNA-seq measurements is probably not the one that you will choose as your favorite one, because yeah, you may be interested on SNPs or copy numbers, but we tend to do more often the single-cell RNA-seq or the single-cell ATAC-seq because it's more high throughput and costs less money to sequence, and it also gives you the regulatory information. So single-cell DNA-seq, as I showed to you, the data is really clean in general, so you can very, very nicely and with a lot of confidence call copy numbers. But we thought, what about single-cell ATAC-seq? How does this data look like? And here what you see is every dot, sorry, this is the HCT116 cell line from a published paper. So every dot here, every green dot is a summary of a small bin in the genome, how many reads are mapping in that position. And the black line is a mean. So what you can see is that you have some regions where the mean is higher. So the mean is pretty noisy, okay? And this below is just like 10 cells where they had done for the same cell line, single cell genomic sequencing. This is not in the same lab, so the cells are different, but these are actually collaborators that were also part of the paper of the ANOFinder first method that we developed. Here we only have like 15 cells, but this would be like the gold standard. So these are indeed the regions that should be gained in that cell line. So what we decided to do is that we decided to develop a segmentation algorithm that will find which ones are these positions in the genome where these knicks are happening between low and high or high and low. And then we would want to call whether a segment has been gained or lost or is deployed. So what we first do, we count the reads in every bin. We, of course, need to remove some blacklisted regions, and then we want to apply a binary segmentation. So this is just a toy data or a small piece of the genome where every dot is a bin. And let's say I want to ask, okay, is it here where there is a nick between the two, between the two distributions, or is it here, or is it there, or is it there? And for example, if it's here, I can say, okay, I split the chromosome into these two pieces, and I ask, are these two distributions the same, or are these two distributions different? And for that, we use the Anderson-Darling distance. And here, for example, it tells us that the distributions are pretty similar to each other, given the cumulatives. But if I find the right spot where actually the change in the distributions has happened, suddenly this Anderson-Darling distance is much different between the two parts of the chromosome. So using that, we identify these breakpoints in the genome that tells us at that position, it looks like the distributions have changed. And then we say, okay, given the mean openness in the genome, has there been a gain or a loss in every one of these segments that I have identified? Okay, so this is what we do. And then we say, oh, look, there has been a gain, for example, in those positions. Okay, so here you see an example of this. This is the HCT116 cell line. We only have 90 cells, and this data set, I must say, it's very noisy because this was one of the first simultaneous profiling data sets that appear, where we have both the RNA and the ATAC-seq for the same single cell. So it's probably not the best data set, but we had the reference genome. That's why we liked to use it. Okay, so what do you see here? Every column is a chromosome. Sorry that the labels are tiny. You cannot read them, but this is chromosome 1, chromosome 2, etc. And every one of these lines is one of these 90 cells. And in green, you can see it here. In green, you have the mean openness, so disomic, in purple, a loss, and in red, a gain. Now, this is the reference that I showed you before, where they profiled single-cell DNA for the same population, although we're not the same lab, so it's not really the same population. What we see is that on our data set, we have a chunk of cells that seem to do not have any gains and losses. They seem to be purely disomic everywhere. But we see that we can recover in nearly all the rest of the cells these big gains that have been identified in the HCT population. We also have noise. You see all these pieces here and there that have been gained and lost only in one cell. But yeah, I mean, you also have the noise if you do the single cell DNA sequencing, which is much cleaner. And even there, you see every one of the individual cells has a little bit of a gain or a loss somewhere else. And what we also see is that there are some of these gains that seem to be common to a lot of cells. So it's most likely not noise anymore, but it's more like a real information on the data. And here, for example, you can see this profile, single cell ataxic profile of one of these cells that has these gains here. And you can see that indeed, I mean, there seemed to be a gain of the arm of this chromosome, for example, or here too. So we don't think that this is noise anymore. We also applied the same method to human data sets of basal cell carcinoma. This is a more recent data set from the 10x platform, so that's why we have so many more cells. We also have a reference for these patients that was obtained using another technique. And this again, every one of these lines here is a single cell ataxic profile for every one of the single cells. Every column is a chromosome. And we again see a chunk of normal cells, which is normal because they also reported on the paper, they had the stroma infiltration, so normal diploid cells that infiltrated the tumor and were also sequenced. We also see these patterns of gains that had been reported for the average of the population, but we also find some level of clonality where there are some groups of cells that seem to also have obtained a slightly different karyotype than the rest of the cells. Okay, so now I will conclude. What we have seen here is that despite these super high levels of noise and missing data that we know are present on the single cell ataxic data sets, it's still possible with a certain confidence to call copy number variations from single cell ataxic data. For doing that, we use this combination of a binary segmentation combined with this Anderson-Darling distance to identify the breakpoints. And what we think, that's why I think it's very nice, is that basically what we are doing with that is that we are adding for free an extra level of omic information on top of any single cell ataxic experiment that you may have done. So this is why we think it's actually a powerful thing, because there isn't much more that you need to do, and then you also get for free the copy number variation calls. So with that, I would like to thank you. And I would like to, of course, thank my group and my collaborators. So on the first project that I presented on EpiScanpy, this has mainly been Anna's project with the work of also Maria and Chris. On the second project, with the collaboration of Fabian's group and at the University of Münster, also it has mainly been Chris and Anna working on it. And the last project on copy number variation, it's the work of Akshaya together with Michael Schubert at ERIBA in the Netherlands. And with that, thank you very much, and I'm happy to take any questions.
Great, Maria, thanks a lot. Totally amazing what you do with that super interesting new type of data. Thanks a lot for the talk. There are questions coming in. I would prefer to call people up so they can ask their questions, so I don't have to read them and we have more of a discussion. You can also raise your hand and then I'll call you from the attendances list. We have over 200 listeners to the talk. Great. Maria, may I start? So we also have a couple of projects where we want to try out a multi-ohm 10x and want to see what we can do with the data. One thing is, of course, to identify cell types, right? That's what people normally do. And I guess having the single cell ataxic information helps in there. So first question, how much does it help? Do we actually need it? How much more information can we gain for that? Second information, cell identity is one thing, right? But cell specification or lineage decision is the other thing I'm also very interested in, right? How much can we gain from that type of data to really understand what process is taking place molecularly?
Yeah, so great questions. Although I come from the epigenetics field, so I would like everyone in the world to measure epigenomics. So when they do single cell, if you are interested in identifying cell types, you're probably going to be able to do it very well with single cell RNA-seq data. So if this is all you need, I would say probably single cell RNA-seq is good enough. So yeah, maybe someone has other opinions, mute yourself and go for it. Single cell DNA openness or methylation is something that is more interesting for your second question, which is more, for example, lineage decisions. Or, so yeah, exactly. For example, what we know is that there are many situations where there will be enhancers in the genome that may become open before the genes start to be expressed, for example. So they are already kind of poised or primed. This is something that you can see if you have the single cell ataxic information. And ideally, and this is something that people have now just started to look at with these true paired measurements, where you can actually see for the same single cells, the same cell at the time, when did the enhancer become open and when did the gene become expressed. So this is where I would say the most power comes from these mixed assays. And I guess this is why, although they are extremely difficult to perform in the lab, everybody's happy to set them up because you can suddenly access all this regulatory information. And I didn't talk about that, but of course, the ATAC-seq data analysis doesn't stop at identifying clusters. What we ultimately want is to know what are the regulatory pathways or landscapes that the cell goes into to identify, to decide are we going to become that cell type or that other cell type.
Thanks, Maria. Now, let's go to the questions. There are a couple of those, so I would all ask you to be quick on questions and answers. Bobby, you were the first. Could you please unmute and turn your video on to have a bit of a discussion?
Hi, I'm Bobby here. Sorry, my video is not working right now. Can you hear me?
Yeah.
Yeah.
I actually have a couple of questions. So the first one, which I posted in the chat, was about feature construction, the feature selection that you mentioned in single-cell ATAC-seq. Is there a way you can incorporate more information rather than just the motor proximity to the gene while assigning genes to genomic loci? For example, high-C data to look at enhancer activity to genes.
Yeah, this is a great question. So at the moment, we did the very, very easy one, which is we go for the closest gene, but I fully agree with you. This is not what we should be doing. We are now actually discussing with some other groups that have done this work of incorporating high-C data and incorporating more like enhancer annotations and make some kind of tool that will search for you, that if you have some coordinates of the peaks that you're interested in, instead of just giving you the closest gene, will give you the gene that is known to be regulated by this enhancer, either because you have high-C data at the bulk level, but for that same cell type, or because there are some catalogs of enhancers which are very well studied. So this is indeed a great question, because going for the next gene is it's a very poor kind of method to use. Agreed.
Bobby, if you have more questions, maybe just write Maria, and so we can go on here and have other people's questions as well. Thank you. Pedro, you're next.
Thank you a lot. It's a really enjoyable talk. My question is about the single-cell ATAC-seq integration. Do the methods, the different methods you compare, use the same peaks combination as you showed for your integration? I mean, you say that, peaks are heterogeneous and you combine them. So do these methods use this peaks definition as you did?
Yeah, no, the methods were developed for single-cell RNA-seq data integration. So they never had to touch the feature space, because they were assuming that you had a gene count matrix, a gene count matrix, and a gene count matrix, the same gene, so easy integration. I mean, easy, no, sorry, but easy concatenation of the matrices. So no, this thing of merging the peaks is something that we needed to come up with the idea. If anyone has a better suggestion, we are very happy to take it. Because it was a lot of discussion also, should we take the union of the peaks, the intersection of the peaks? Should we take the most variable peaks before or after integration? Should we? So yeah, it's probably not ideal either, but it seems to be working very well. So it seems to be working best at the level of integration. So
yeah, thanks. Maybe it helps that you see for some patterns in the different methods you compare.
Mm-hmm. Good point. Thanks.
Next one, Mark.
Yes, thank you very much. So I have two questions also. You focus on data integration of single-cell data, but there might be good reasons also to integrate bulk data and single-cell data, maybe like bulk methylomes and single-cell methylomes, for example. This is what we are interested in for different reasons, data availability, or for example, applying plate-based single-cell omics where you have low numbers of cells and so on. So can you comment on this? if you also are able to do this with EpiScanpy, for example. And my second question would be regarding evaluating data integration. So very often, very simplified spoken, a readout is how well data is overlapped of different data sets is overlapping. So the question is always there with real biological data, if this might be also artificially enforced. So how can you control for this? For example, we know, especially with chromatin regulation, the different wild-type mouse strains, for example, can be quite different in terms of chromatin regulation. So maybe you can also comment on this.
Yeah, thank you very much. The first question, I must admit, we don't do it on EpiScanpy, and it's not something that I had really thought about. It's a very good point. I guess what one can do at the level of batch correction probably would be using your single cell pseudobulk and applying to that data, the pseudobulk, some batch correction that would be for bulk data with your bulk DNA methylation, for example. But then you would need to go back to the corrected single for the pseudobulk and try to deconvolve it again to get to your single cell information. So I wouldn't know, I mean, I'm sure one can think about how to best do it. For the second question, yeah, great question. So, I mean, on the paper, and Malte has his camera turned on, and Malte is the first author of this benchmarking paper. Malte, Marin, and others put a lot of work on coming up with a very long list of metrics that we could use to assess both indeed how well mixed the batches are, both at the level of the embedding, but also at the level of the KNN graph and so on, where we really wanted to know how well mixed they are, but how much biological information we have. And for example, on this ATAC-seq data set that I showed the integration, there was, for example, one cell type that was only unique to one of the 3 data sets. So we also looked at some metrics, for example, the isolated silhouette or things like that, that told us if I have a cell type which is unique to one data set, but I asked my methods, my integration methods to integrate the 3 data sets, will it by default try to integrate that unique data cell type with whatever that it's a little bit similar to it or not? So this was also kind of taken into account into the results of the integration.
Very good. We're a bit over time already, so please, the remaining 2 more questions very briefly. Malte.
Thanks a lot for an interesting talk, Maria. I especially like that second topic. It was great. No, so I actually have a question about the last part. I really like the kind of the copy number variation inference, but you showed this on the level of karyotyping, right? So you showed this on the chromosome level. I'm guessing it'll be most interesting for analyzing kind of patient data on the gene level. How sensitive are you and how far down can you go into that gene level when looking at kind of patient data rather than immortalized cell lines, probably, right?
Yeah, so we cannot go down a lot. The beans are now 100,000 base pairs. So this is kind of our resolution. It's very broad. What we are going to do next, of course, is that for the HCT cell line, although the data was very noisy, for the same cells, we have the RNA-seq. And then what we can do, for example, is that we can say, okay, I have this large area that has been gained. So it's large indeed, but I also have the gene expression. And at every one of these kind of subclones that we have seen, we always have a group of cells that were disomic versus a group of cells that were trisomic. So we want to check what happened actually to the gene expression on those regions. Do we also see an increase? Yeah, it's large. It's far from the single gene resolution. It's more like at the arm level. No, I mean, we can see rather small copy numbers, but not at the gene level.
Okay, thanks.
There's a final question from Pau.
Yeah, thank you so much. I saw that you compare EpiScanpy to other methods, but have you benchmarked it against Archer?
No, no, it's just appeared yesterday or last week or 2 weeks ago, isn't it? It was on bioRxiv for a while. We didn't compare it to it. Archer, oh, come on, remind me, there have been a lot of pipelines appearing lately. What does Archer use for clustering? Sorry. Is Archer using LSI, LDA? I cannot remember anymore. Maybe LSI?
Pau, this goes back to you.
I actually don't know. I'm Yeah, I have no idea, sorry.
Well, I may be wrong, but I think Archer, it's from the Kuzanovich group, and they use LSI, which is the same method that was used initially on the Kuzanovich et al. Paper. But I may be wrong. Don't, please, this is being recorded, but LSI, if that's the case, then LSI would be the same method that was used on the Kuzanovich et al. Paper that was part of the methods that we compared it.
Great. I guess
and directly compared to it.
It's still in bioarchive, I guess, right? The manuscript.
Yeah.
No, yeah, maybe try that out. Congratulations to Nature Communications, and thanks again to Maria for this amazing talk and all the attendees and great questions. I'd like to finish by saying that you should be here in 2 weeks again, same time, same place. We'll have Stefanie Grosswend from MDC Berlin talking about the roles of polycomp in embryogenesis. And again, it's about single-cell transcriptomics. Surprise. Thanks, everybody. Have a great Friday and a great weekend. See you the latest in 2 weeks. Bye-bye.
Thank you very much. Bye.